thank you for the introduction. And so to be honest, I'm not very psychologically prepared for live speaking event, but yeah, too late to be nervous, right? So let's get to it and let's see how it goes. So yeah, as he said, like my name is An-Phi. I'm a PhD student in IBM research and ETH Zurich. And today I'm gonna talk about DECODE, our computational pipeline for basically for understanding T-cell receptor epitope binding rules. And this is joint work with Ileana, Anna, and Maria, my three main collaborators on this project. So let's have a quick look first at what's the biological motivation behind our project. And so if you have been living on this planet before, right, one thing that you might know is that we actually are continually exposed by a high number of antigens that try to attack us and makes us sick, let's say, right? So one of the defense mechanisms that we have against these antigens is the adaptive immune system, right? And one of the core components of the adaptive immune systems are the T cells. So T cells are these, cells that basically adapt in a way to recognize the specific epitopes, antigen epitopes, right? And throughout our lives, we build this T cell repertoire. And this T cell repertoire is a very important source of information from a clinical perspective, because you can actually understand the whole immune history of a patient if you can understand this this repertoire, right? But like we have two main problems here. And the first one is that we don't actually know which T cell is going to bind to which antigen epitope. And the second one is, even if we can predict this, then we don't actually know the underlying biological mechanism to understand like the driving the binding of the T cell receptor and the recognition of the T cell receptor of the epitope. So, this work is actually focused on this part because, to be honest, in the last years we have been seeing, thanks to the increase of data and like the successes of deep learning, for example, we have seen many models being developed for this particular task, right? So, and the thing is, because they are trained on different data sets, or they have maybe different inductive biases because of the modeling of the architecture, then most probably these different models will give us different insights into the T cell receptor binding problem, right? And the other thing is that not all models have some interpretability capability to tell us what are the binding routes that are driving this phenomenon. So what are we proposing here in this work? It's basically just this computational tool, that wants to help people working in this sphere to understand any kind of TCR epitope prediction binding model, right? And we want basically the two core things of this pipeline is that, again, this can be applied to any model that you are playing with, and it wants to give you basically a global overview of, so a summary of what could be these rules that the model is using to predict the binding. So a quick note first. Of course, this is not the best way to do it. It's just one way to do it, right? So we're not claiming anything in terms of performance on the interpretability side. Right? But what we're focusing on with this pipeline is really from a user perspective. So we want a pipeline that is a generic, readily usable, customizable, and tunable. And we'll go into the details a little bit later about this. So to summarize, the real goal of this project is really to enable any user and any researcher of T-cell receptor epitope binding to have a quick look into any model that is trying to solve this problem. So basically enabling them to have a first interpretability step into their problem. And yeah, so let's jump a little bit into the details. Like I'm gonna give you a quick overview of how this pipeline works. So there are four main parts: initial pre-processing, and a final visualization and a post-processing of the results. But the two main core components of this pipeline are these two, like a clustering step and an interpretability step. So looking a little bit into the details of the clustering step, so this is the step that is going to give us the summarized overview of what the model is doing. And the idea is simple. The concept is very simple. Basically, what we want to do here is summarize the data set that we're working on with a few prototypes. And by running later the interpretability step on the prototypes, we're going to have like a global overview of what the model is doing. Then a little bit of details. So our pipeline actually implements already some algorithms that are well established, like dbScan. Of course, there's k-means, k-medoids, and so on. And the other thing is that like maybe you don't know which algorithms you want to use. So what we allow here in the pipeline is to define to tell the pipeline to use multiple clustering methods, and the pipeline is going to run all of them. And the user can define a score to evaluate these clustering methods, and the pipeline will tell which one performed best according to this metric. Then let's have a look at the interpretability step. Not sure if any of you is acquainted with the field, but so what the interpretability step is doing is basically just using a very established algorithm called anchors. And what it's doing is basically taking a sample, so one of our prototypes, and then from these prototypes try to extract the binding rules. So like why this prototype was classified in a certain way, either binding or non-binding, right? And the rules basically can be understood in this sense. So basically it's gonna say, if a certain amino acid is in a certain position and another amino acid is in another position and so on, then like the sample is gonna be predicted by the model in this way. So like a very quick look of how anchors works. It's basically just a perturbation analysis. So the algorithm is just gonna change the amino acid of your sample, your prototype, one by one, and increasingly with groups of amino acids, right? And it's gonna do this using some dynamic programming algorithm to make the optimization a little bit faster. So these were the details, and let's have a look now at the four claims that we did before, right, about our pipeline. The first one being generic and readily usable, right? So this is the main part, because you want to be able to use use our pipeline on top of your model that you want to understand and interpret, right? So the two core things that you can do here, it's define your own model and your own data set. And this can be done in a relatively easy way, I would say. So basically, you just need to define your model in a model.py file, your dataset in a data file, and then put them in the same folder. And then you run the command line for running the pipeline, right? So you do this by basically, you can see the cursor, hopefully. So you just point the pipeline to where you define your own model. And then we have the tunable part here, where we're gonna go a little bit into the details later. Then let's have a look at the next claim. So it's customizable. Well, of course, if you can apply to your own model and your own dataset, it has already some degree of customization, right? But on top of that, you can actually define your own clustering method, because maybe you want to try something more cutting edge, or maybe there is some clustering method that we have not implemented but works better for your data set. That could be. And then you can actually include that in the pipeline. And on top of that, you can perform more pre-processing, because as we know, like not all the clustering methods work on all the data types that we could have. And this can be done basically in the similar way as I shown before. You put all the methods in a file called clustering.py, surprise, and then you put it in the same folder. And then it's easily tunable, right? So this part is basically So as you know, like clustering algorithms have many hyperparameters, and to be honest, also anchors is sometimes difficult to tune correctly, and maybe your model might have some hyperparameters as well at inference time. I don't know, but so the thing is that like one way to do this would be to just hard code the new values in your code, but in our pipeline this is not necessary. Like we provide a text configuration file, a template where you can modify all the parameters that you have, and you just pass this configuration file to the pipeline, and it will run it with the hyperparameters that you want. Yeah, I can skip this. So now the question is, like we have a bunch of hyperparameters, a bunch of different results or explanation for our model. Then like the question is, how good are these explanations, right? And the thing is that we don't know. So this is actually a pretty big topic in interpretability. And I'm not sure it's gonna be solved soon or if there is any solution. And the core thing is that like good is subjective, right? Now we don't know, we don't have an objective measure of what a good explanation is. So what we actually provide then in our pipeline is just a set of metrics that can maybe guide you as the user in trying to understand if the explanations are sensible or not. And like some metrics that we have are, of course, the metrics for evaluating the clustering. For example, the silhouette one, we will see it in an example later. And these three metrics that I'm not gonna go into the details now, but like you can look at the paper, but like the two main things that they try to measure are how general the explanations are. So in a way, like how the knowledge that you get out of the pipeline is transferable to another data set, for example, or to some other samples, and how complete the explanation are. So basically how comprehensive this set of rules that you extract are how comprehensive in summarizing your model. So let's have a quick look into an example. So here we're just gonna run the pipeline on Titan, which is a model for a T-cell receptor epitope binding prediction developed by one of my collaborators that I showed before, Anna specifically. Okay, data is not too important. It's just a subset of the VDJ database in case you're familiar with it. And this is an example of a visualization that you could get from the clustering step. Where like here, for example, we're running 3 different algorithms with each with five different set of hyperparameters, and we're asking the pipeline to evaluate it with the silhouette score, and then like it's gonna detect that particular one agglomerative clustering with certain parameters. And on top of that, what you can also do is ask the pipeline to show like a low dimensional projection of the cluster, just to have like a visual qualitative analysis of the results. And this can be done with t-SNE and UMAP. And actually, here I didn't mention, you can customize this part as well. So you can add your own dimensional reduction algorithm if you want. And then this is an example of the interpretability step. So one of those clusters, like if we take the sample for from one of those clusters, then we get out this rule. Again, like I'm not gonna go too much into the detail because this is just for demo purposes, let's say, and we haven't really looked at the biology yet. We haven't validated yet. But so the like two quick comments that we can make on this are that, as you can see, so in the lower figure, you can see like just a motifs, just a logo. So basically, a frequency counting of the amino acid in your data set. And on top is the actual rule that the pipeline has computed. And in this case, you can see that the pipeline is not, well, basically anchors is not just picking the most frequent ones, which is nice, I guess. And then it seems in this case that for this particular sample, well, I don't remember exactly which epitope we're trying to bind, but it seems that like the T-cell is using this particular V segment for to predict the binding. And yeah, I'm gonna skip this. So yeah, we're at the end. of this exciting talk, I would say. And yeah, to summarize, yeah, we're proposing DECODE, a computational pipeline to try to interpret any TCR model. And again, like we're not providing this solution, but a solution that that tries to be user-oriented, right? Like so generic, ready to use, customizable, and easily tunable. And again, like the goal is really just to enable you as the user, a researcher, to basically have a first look at what your model is doing or the model that you're interested in is doing. Now, let's have an open heart moment for a second before actually concluding. So to be completely honest, like I think there is a little bit of a steep but short learning curve in trying to use this pipeline, in a sense that like I just quickly went through it, but the configuration file is actually quite big to take in at the first sight. So you might have to try to play around and see how everything works. But like, according to some colleagues, it seems like that after you do that, like it should be fairly easy to play around. And the other point is that the visualization that you have seen, in the current version of the code, like we actually decided to take them out because we actually seen that visualizations are more useful if they can be interactive with the user, right? So, and this is a little bit difficult with the pipeline because it's in a certain way it's static. So for now, we're providing some boilerplate code that we're gonna clean a little bit more and then push. But what we would like to have at the end, it's a separate standalone component where there is we provide this visualization in a more interactive way. And yes, and to be completely honest, I'm not a UX researcher, right? So I think this is an easy pipeline to use. Some colleagues claim the same as well, but of course we're not the only. We don't want to be the only user, right? So this is where you come in. So if you want to use it and you want to give us some feedback, we would appreciate that. It would be great. And yeah, so you can follow that QR code. Again, to summarize, in the future work, we want to improve the user friendliness with this interactive visualization. We maybe want to add also a framework to automatically optimize all the hyperparameters that I mentioned. And maybe we will see if it's really useful, maybe a wrapper, like an interface to R, because R is pretty common in bioinformatics, so maybe it could be useful, but we will see about that. And yeah, as a final point, anchors might not be the best to use as an explainability method, and we will evaluate if adding more or maybe just substitute it. Yeah, to conclude, I would like to thank my three main collaborators, Ilyana and Maria, my PI, and yeah, all the other people from our computational system biology group, and IBM research, of course. I mean, I have to say it, right? And like our funding body. And yeah. Yeah.
So the variability of the CDR3 is much wider than of the VGENs. And if you use Anchor or anything similar like that, there is a way better chance that you'll find the cluster on the CDR2 and CDR1, which are easy to find because it's enough to have some bias in V, and you're going to miss the main signal, which is the CDR3.
Yes, so like that's a very good question, and like we did receive that during like the review phase, right? And the thing is that okay, like it's a complex mechanism. Like we would expect the rules to be mostly in the CDR3 region, but we also don't know. Like also the other things might be important, but in this case, so it depends on your data set. Of course, you can also decide to shrink everything in the CDR3 region, and it will show you if we show you only the rules in the CDR3 region. That's possible, but in this case, for some reason, the pipeline told us that like the CDR2 was more important, and there the question is, what is wrong? Is the model wrong? Is the pipeline wrong because of the hyperparameters? We don't know, and that's where the tunable part comes in, and it's important to play around with.
Hi, Thanks for the talk. I had the question about the application of this pipeline. So when I create a model and it does the prediction and I run your pipeline and I get the output, how do I use that then, or for what reasons do I use that?
Well, it depends on what you're looking for into your data, right? So maybe, again, like maybe you want to know which specific amino acid seem important in the CDR3 region. And if the model, if the pipeline tells you that there is something important there, then it's up to you how to interpret, right? So with interpretability here, I just mean that like we create the explanation. Then of course, you as a researcher have to decide if it makes sense. So if maybe it is because again, like the model is just doing crappy things, or maybe there is some biological significance there. So, and then like from there, if you think there is biological significance, then you go on with the experimental validation and see if it actually makes sense. And yeah.
Okay, thank you.
And in 30 seconds, there's a question online about whether this can be applied to model BCR binding.
Sorry?
To apply to model BCR binding.
Yeah. Yes. So like, I mean, like this is just our application, but so I think like any text-based sequence model that you can apply to.